Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Aina binti Wahabi, number matric S72469. So, today I will present about optimization of essential oil extraction from leaf of ananas squamosa. So, in this assignment, my supervisor is Dr. Ahmad Nazis bin Aziz and Dr. Faizatushima binti Muhammad. So, I will explain about research background. Essential oil is a concentrated liquid containing volatile and chemical compound from many plants. Essential oil, also known as volatile oil. Essential oil can be extracted from leaf, fruit, seed, stem, flower, and root of the plant. The essential oil can be extracted by the process hydrodistillation. The essential oil have gained their importance in therapeutic, cosmetic, aromatic, and fragrance. This is because essential oil is formed by volatile compounds such as terpene, sesquiterpene, and several oxygenated derivatives. The use of essential oil in Malaysia has become a major attraction because this essential oil widely used in daily life. However, there are also essential oils from plants that are less explored, such as Ananas squamosa, which has not yet been fully explored, especially in terms of chemical composition. So, in this research, we can know how to get essential oil from the Ananas squamosa. The problem is availability and quality of raw material. The availability of Ananas squamosa in Malaysia is low compared to other countries. Another problem is leaf of Ananas squamosa for essential oil production. The small leaf of Ananas squamosa that limited will be effective for it to produce essential oil. Lastly, the extraction takes a long time, so it leads to degradation of active compound. So, the research objectives: firstly, to know the method of extract of leaf of Ananas squamosa to get essential oil. Secondly, to know how to obtain it better in essential oil from leaf of Ananas squamosa. Thirdly, to know the chemical composition in essential oil from leaf of Ananas squamosa. Lastly, to know biological activities of essential oil from leaf of Ananas squamosa. There are several scopes that have been identified to study. First of all, size of sample and quantity of the sample might be better in yielding of essential oil of Ananas squamosa. In addition, to study effect of time conduct the extraction process. The focus of this study to observe the optimum time needed to obtain essential oil of Ananas squamosa using hydrodistillation technique. Next, to study about the gas chromatography mass spectroscopy TCMS to analyze the component in essential oil. Lastly, to know biological activities of essential oil of Ananas squamosa. Because of it have many compounds, it allows this essential oil to be antibacterial and antifungal. Because of these properties, it will this impact for our economy. This essential oil can be used in industries such as cosmetic or pharmaceutical. So, I will explain about the background of Ananas squamosa. Ananas squamosa is known as a custard apple or sugar apple. It belongs to family Ananaceae and is important fruit in West India, South America, Peru, and Mexico. The tree of Ananas squamosa is small, about 3 to 8 meters. Next, the fruit is spherical conical, has 5 to 10 cm, which is 2 to 4 inches. The weight is 100 to 214 grams with a thick ring composed of knobby segment. The color of the fruit is pale green with a deep pink flush. The seed of Ananas squamosa around 1.3 cm to 1.6 cm, which are brown to black color. Moreover, Ananas squamosa has history in traditional medical. For instance, the dry leaf on Ananas squamosa are used for analgesic effect. Other than that, the root of Ananas squamosa are used for dysentery medical. Next, leaf of Ananas squamosa is used to treat the ulcer. Scientific research has concluded the fruit have many benefits based on their traditional use. So, I will explain about chemical composition in essential oil. The chemical compound in essential oil typically have hydrogen, carbon, and oxygen. It can divide into two main categories, which is hydrocarbon and oxygenated compound. For hydrocarbon, this only contain hydrogen and carbon. This group is made up from terpene, monoterpene, and sesquiterpene. Secondly, for oxygenated compound, it have carbon, hydrogen, and oxygen. For example, ester, aldehyde, ketone, and phenol. The bioactivities of essential oil of Ananas squamosa, firstly, is antibacterial. Essential oil of Ananas squamosa will inhibit the bacteria due to the present bioactivities compound like phenolic compound, alkaloid, and terpenoid. Secondly is antifungal activity. The essential oil from Ananas squamosa are equally impressive. Antifungal activity can promote for managing fungal infection in human and plant. There is anti-cancer activity. Essential oil of Annona squamosa are anti-cancer because have hexogen that known as cytotoxic effect. This will inhibit cell cancer by targeting their mitochondrial complex. For characterization of essential oil of leaf of Annona squamosa constituent, it can be carried out by basic retention index Ri by calculate using homogeneous series of alkane and by comparing the mass spectra and retention data. In gas chromatography, many different types of strain used for making sample injection. The injected sample should not be large in size and should enter the column. Next, for column selection, there are two types of capillary, which is capillary column and packed column. To use for essential oil that contain volatile compound, capillary column are most efficient separation than packed column. This is because capillary column have higher resolution than packed column. Capillary column only need a small sample, meanwhile packed column have low resolution and need large amount of sample. Lastly, for detector used in a gas chromatography, firstly flame ionization detector, thermionic emission detector, or mass spectrometer. For methodology, firstly, we need to prepare the sample. The plant material of Annona squamosa will be collected. Next, we need to prepare the chemical and apparatus. After that, we need to set up apparatus for hydrodistillation. So, we need to set up the hydrodistillation apparatus like this picture. The plant material of Annona squamosa is treated into small pieces and transferred to a round bottom flask. Add the plant material not more than half full. Water is added to approximately half in the round bottom flask. Heat the plant material using Bunsen burner until the water in the flask boils and it can generate steam. After that, the distillate mixture of oil and water can run from condenser to the conical flask via the receiving flask. The distillation will contain water and oil. After collect the distillate in the receiving flask, the distillate will undergo extraction. Next step is add distillate into separating funnel and add hexane. Aqueous layer will be extracted for second time. After extraction, combine the organic layer and magnesium sulfate and filter the solution. After that, we can weigh the essential oil and place in closed and glass. After that, the essential oil will undergo gas chromatography to characterize their constituent. So this is my expected result.